investigator comes to us and they begin to present their project and what they're trying to do, my first question is always the endpoint. I like to start with the patient. Who's your patient population? What condition do they have that you're trying to treat? And then we can back off from there and start talking about, okay, what's the formulation of your final product? Is it a pill? Is it a dose of cells in suspension? Is it a flat tissue, like a skin treatment? Is it a solid organ? What are they trying to produce? And then moving on from there, how is this treatment applied? Do they take it twice a day? Is it a single implant and you've provided a cure in one dose, as we like to say in regenerative medicine? So what's the final formulation and how do you want that applied? We have GMP manufacturing engineers. Quite often we hire biomedical engineers into these positions because we want them to be very intelligent, critically thinking, mechanically skilled people doing this kind of work. But you have to assume that this person knows nothing about what you're trying to do. And so when you translate that technology, you have to be able to spell out every single step in the procedure. And we put these into what we call batch records. So a batch record is literally a step-by-step accounting of the procedure that you're going to be following, places to record any data, any calculations that you do along the way. And each operator who performs this step will initial and date that they have performed it, and you will always have a second operator in the room with them watching what they're doing and verifying that every single step was done. And so there's this concert, this coordination between the two operators in performing this procedure. And that type of process can be really foreign to a researcher who's simply used to sending the technician into the lab to do the procedure. The environment that they work in is also very different, and you have to take that into account when you're talking about translating a procedure from the open lab bench into a GMP cleanroom facility. You are in a GMP cleanroom facility to control personnel movement, to provide clean surfaces and clean air that you can perform this procedure in. And that's much more advanced than just a sterile hood where you put your hands in the hood and assume that's going to provide the sterile environment for you. So procedurally, these operators are trained to only move in certain ways within the entire environment. And the entire environment that they're in is an enclosed space, and they're typically gowned head to toe, face mask, hair cover, full sterile gowning, so that the operator doesn't provide any contaminants, as best as we can with the risk assessment that we've done, to contaminate the product. So scaling up can be considered in two different ways. So one, you've treated a mouse and now you want to treat a human. Does the dose scale up to a human-size application in the same way that it would number of cells per kilogram? Does it have the same effect, the same result? Does it provide the same treatment in a human that it would in a mouse or a rabbit or non-human primate? So that type of scaling up is something that has to be worked out also. And quite often you just simply don't know until you're in that phase 1 trial, did this dose have an effect? And of course, the phase 1 trial is primarily about safety. So it may or may not have an effect in phase 1. If you have no effect, then most people will pause and go back to the drawing board and say, okay, our product was safe, but it had no effect whatsoever in phase 1 trial. You're probably not quite ready to move on to phase 2 because you haven't figured out the dosing yet or the application precisely. The other part of scaling up large scale means you're doing multiple doses for multiple patients. And so on the lab bench, maybe you've treated one mouse or 2 or 3 mice at a time. In GMP manufacturing, the idea is that you're going to be treating multiple human patients. So in a phase 2 trial, you could have as many as 100, 200, up to maybe 500 patients, even in a phase 2 trial. Have you ever made that many doses of your product, even on the smaller scale? And what will it take to produce that many doses? And as you move up through the phases of clinical trials, you have to think more and more about automation and things like that. So it's typically very manual, especially in phase 1 regenerative medicine applications. One process for one patient may take half a dozen people a month to produce that product for one patient. So how are you going to do that with 100 patients? Is it the same process by the time you move up to phase 2? And so those things, the earlier you start thinking about scale up, both to the human size application, the human safety level, and the number of patients that you're going to be needing to treat, both in the clinical trial phase and later on when this becomes a licensed product, you could be treating millions of patients. Is that the same process that you walked into that phase 1 trial with? The types of things that I would speak to are who controls what part of the process when you begin to work with the CRO. The more that you can take care of up front and hand off in a ready-to-go, ready-to-produce product, the lower your costs will be. Time consideration, once again, the more you can work out up front before you hand it off to a CRO, the more quickly they will be able to reproduce your process and your product. So those are the types of things that I would consider. And, once again, what testing will they do? Do you determine the testing, or do you work with them to let them determine the quality testing? At the end of the process, do they produce a certificate analysis, and what do those release tests look like that you put on that certificate analysis that verify purity, potency, safety, sterility, and characterization of your final product?